This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 258, recorded on January 6, 2022. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schechter.
Howdy. Nice to be here.
You too. Also from Charleston, South Carolina, Michael Schmidt.
Happy New Year, everyone, and happy birthday a few days late to you, Vincent.
Thank you, Michael. We're moving on in years. Can't stop it.
No, time and tide waits for no one.
Our first TWiM of 2022. Happy New Year, everyone. And here's to maybe 24 this year. So we did last year, right, Michael?
Yeah, this is our 11th anniversary that Vincent, I, and Elio have been doing TWiM.
Wow.
11 years.
My goodness.
Yep. Time flies. All right. Today we have a snippet in a paper. As usual, I will lead off with the snippet. Very interesting paper published in Cell, Host, and Microbe. Symbiont-regulated serotonin biosynthesis modulates tick feeding activity.
Who would have thought?
Who would have thought? I guess we should have. By Zhang, Peng, Zhao, Wang, Tang, and Wang from Fudan University and Fudan University, all Fudan University in Shanghai. And this is all about ticks and their symbionts. As you know, ticks can carry infectious diseases. And
plenty of them.
Plenty of them. And at each stage of development, the larvae, the nymph, the female, they need a blood meal before molting or laying eggs. And this is the thing I didn't know. Ixodid ticks spend days to weeks on their hosts. Days to weeks taking a blood meal, and they ingest blood equal to 100 times more than their body mass. Oh my gosh, I had no idea they would spend so much time. So
this is why it's so important to get a tick off of you, because they're transferring microbes to you at the time, and Ixodes ticks, transmit Lyme disease.
That's the point.
So they're spending a long time on the host, so they have to do a number of things. They have to evade host defenses, and they have to make sure they can take blood so it doesn't clot. So their saliva contains hundreds of bioactive molecules, vasodilators, anticoagulants, immunosuppressants, and they're also painkillers so the host doesn't feel them and let the blood flow. And in fact, the midgut, which takes the blood, that's where it's stored and digested, has various molecules involved in detoxification, proteolysis, nutrient assimilation. So tick blood feeding, as they say, is very complicated, and we don't know much about it, frankly. But what we do know is that ticks have symbionts, and the common ones are bacteria of Coxiella, Rickettsia, and Francisella genera. And in fact, as you might gather, these symbionts are essential for tick development. They provide vitamins and cofactors that aren't very common in mammalian blood. And of course, in turn, the symbionts rely on the tick to get nutrients needed for their growth because they can't provide everything themselves. Now, furthermore, it's thought that the symbionts are somehow involved in regulation of energy balance in the tick. So in this paper, they wanted to look at the relationship between the symbiont and the feeding activity of the tick. And this is going to involve some molecules, including 5-hydroxytryptamine, or serotonin, and a variety of enzymes involved in its biosynthesis, as well as tryptophan. So the first set of experiments they do, so the cast here is the tick, the Asian longhorn tick, Hemaphysalis longicornis. Okay, and its symbiont Coxiella species. And the first, so Coxiella is the major symbiont in H. longicornis. And so they said, what are these symbionts doing? So they treat the ticks with tetracycline. They treat nymphal ticks, and they inject it into the tick. And at 0.2 micrograms per nymph of tetracycline, it reduces the abundance of the Coxiella 2 or 4 days after treatment. But it doesn't affect the rest of the microbiome, interestingly. That's a good choice of antimicrobial. And then when you allow the ticks to attach to a host, the tetracycline treatment blocks the attachment. And the ones that do attach don't take up much blood compared with untreated ticks. And the tetracycline-treated ticks have fewer Coxiella bacteria. And so this was the first clue that somehow Coxiella plays a role in blood feeding. So you take the Coxiella out of the tick, they don't do a good job blood feeding. They don't even attach, and the ones that do don't drink very much blood. I wonder if you could treat vampires with tetracycline and do the same thing.
I am sure that was the intention of Anne Rice when she was writing her vampire novel series.
Right. So then they treat the ticks with a mixture of antibiotics, penicillin, streptomycin, and gentamicin. Doesn't affect Coxiella, but as you might expect, it decreases the microbial load in the tick, and those ticks still ingest the same amount of blood. So it's the Coxiella that is involved in somehow regulating tick blood feeding. So in this modern day and age, what would you do next? You do a gene expression profile, of course, and then you get your clues from that. So they take the nymphs that they've treated with tetracycline, with and without, and they do RNA sequencing. And of course, you can see different pathways of genes impacted, and 6 that they noted here were involved in amino acid metabolism: tyrosine, tryptophan, phenylalanine, histidine, glycine, serine, threonine, and arginine proline metabolic pathways, which tells them maybe the Coxiella play a role in regulating the tick amino acid metabolism. And then they looked at the genomes of both the tick and the Coxiella to get some clues. And in fact, in Coxiella, there is a complete shikimate pathway, which is used for chorismate biosynthesis. And chorismate is the precursor of aromatic amino acids, which would include, those are the ones with the rings, right?
Mm-hmm.
Tryptophan, phenylalanine, and tyrosine. So they're thinking maybe if you reduce the number of Coxiella in the ticks with tetracycline, you get less chorismate, and then you get less aromatic amino acids, and maybe that decreases tick feeding activity. So that's a hypothesis, right?
That is indeed.
So let's test it. They give ticks tetracycline, and then they give them different amounts of chorismate, right? Chorismate, the precursor to aromatic amino acids. It restores the blood intake of ticks treated with tetracycline. Perfect experiment. I love it.
It was so elegant in its simplicity.
And then they add individual amino acids.
Oh, yeah.
The aromatics. Only tryptophan promotes tick feeding when you treat the ticks with tetracycline. Who was the guy who did the tryptophan operon? Was that Charlie
Yanofsky? Yeah.
Okay. So let's explore this further. In mammals, most tryptophan is metabolized through the kynurenine pathway, and about 1% is metabolized through the 5-HT, the 5-hydroxytryptamine pathway, which is also serotonin. Serotonin is an important biochemical everywhere. Specific.
But here it's going to tell the tick whether or not it needs to eat.
That's the idea. We don't know how it does it, but serotonin has all kinds of neurological and behavioral effects, and so presumably that's what's happening, So they do a series of experiments, and it turns out that Corismate and the downstream metabolite 5-HT are responsible for modulating tick feeding activity. So it's the 5-HT pathway, which is a minority of the metabolism of tryptophan that's really involved in tick feeding. And 5-HT is serotonin, so that makes perfect sense, right?
Mm-hmm.
Okay, so now they say, let's focus on 5-HT, serotonin, and make sure this is involved in tick feeding. So they can inhibit the biosynthesis of serotonin. There's an enzyme, tryptophan hydroxylase. It converts tryptophan to 5-hydroxytryptophan. That's 5-HTP. It's the rate-limiting enzyme. And so they suppressed its activity by microinjecting a double-stranded RNA, which will inhibit the mRNA and downregulate the enzyme. They can also treat ticks with a TPH antagonist. Again, that's the tryptophan hydroxylase, an antagonist, and both treatments suppress tick blood intake, right? So looks like tryptophan hydroxylase is an important control point here. They also look at the receptors for 5-HT, serotonin receptors, right? And they can knock those down. And in fact, it has the same effect on blood feeding. If you knock down the receptors for serotonin, it decreases blood feeding in the tick. So quite clear, serotonin is the key molecule here.
And they have an easy assay that just asks how much blood is consumed.
It's very easy, yeah. All right. Now, corismate is, as I said, the precursor for aromatic amino acids, including tryptophan. The genes that convert corismate to tryptophan are not present in Coxiella, and they're not present in the tick genome.
So guess what? Guess what, folks? We have other microbes.
So they're hypothesizing that corismate promotes serotonin production by modulating the biosynthetic pathway instead of increasing the synthesis of tryptophan, right? Because those genes are not present. And so they do the series of experiments to address that, which, in the interest of time, I won't go into. But they find that corismate-mediated elevation of serotonin does not occur through regulation of TPH because those genes are not present. Now, there's another enzyme, AAAD, which is aromatic amino acid decarboxylase. Okay, it takes off the carboxylate from aromatic amino acids. And so that is responsible for the conversion of 5-HTP to 5-HT to serotonin. So they do some experiments to look at AAAD, and in fact, they find AAAD expression is upregulated by corismate directly. So corismate upregulates AAAD, and that promotes the synthesis of serotonin. So that gene is there. So that makes perfect sense. Now, there are two major pools of serotonin in mammals. There's neurological brainstem serotonin, and then there are Malpighian tubule serotonin. And so they would like to know which pool is involved here. And it's basically a reduction of, so Malpighian tubules is actually where the Coxiella reside in the tick. And it's a reduction in them, in the Malpighian tubules, that decreases serotonin in the midgut and the CNS. So it's the Malpighian tubule is the place.
And that's where they get the correlation that it is indeed Coxiella that's responsible for the increased serotonin.
That's right. And they also do a series of experiments that tell them that Coxiella remotely controls biosynthesis in both the syn ganglion, the nervous system, and the midgut by secreting corismate into the hemolymph, and then it can move systemically. And then serotonin in the midgut and in the syn ganglion then regulate the blood ingestion by the tick. How that happens, we don't know in this paper, but that makes perfect sense because serotonin has all kinds of activities. And finally, very interesting last finding here. So shikimate pathway, right? This is present only in bacteria. It's not in ticks. They said maybe if we could inhibit it, we could inhibit tick blood intake. Glyphosate Sound familiar, folks?
Oh, yes. Roundup.
Roundup, a very commonly used herbicide. It inhibits 5-enolpyruvyl shikimate 3-phosphate synthesis, which is required for the synthesis of chorismate. I guess that's how it is an insecticide, right? Or
yeah,
it's an herbicide, isn't it?
Yes.
So glyphosate inhibits an enzyme that you need for the synthesis of chorismate. So they treat their tick nymphs with glyphosate, and that inhibits blood intake.
It's incredible. The herbicide is used to control ticks. But, that herbicide has issues that people are looking at it carefully. But you think about, a spray that you might spritz on yourself after hiking that could actually, even if you have occult ticks that are hidden someplace, like in your dense hair or, somewhere that you can't see, if you just spritz it on yourself, that would actually inhibit this pathway. The tick would naturally fall off and you wouldn't be worried about mouth parts being left behind and transmission of bacteria, etc., into your system.
Yeah.
So,
but of course, Roundup is very bad. So,
Roundup's bad. But if you could somehow just inhibit this with some other mechanism,
sure.
I mean, just imagine if you could make a CRISPR-Cas molecule that would be cheap enough to spritz on.
Did you say?
Yes, CRISPR-Cas. It's the universal solution to everything, Elio.
But now we have many bottles of Roundup in our garage that we're disposing of because we don't want to put it in the environment anymore.
No,
it's unfortunate because it works really well.
Oh, yes, it works really well.
Anyway, so that's the paper. Let me summarize. So Coxiella regulates tick blood feeding by influencing the synthesis of serotonin. And it is chorismate from the Coxiella, the precursor of tryptophan, that stimulates the expression of an enzyme that leads to the synthesis of serotonin. And so then the serotonin somehow influences tick. It stimulates the tick to feed on the blood, and how it does that, we don't know. But that's neurobiology, I think, not microbiology. So they make an interesting statement here. The Coxiella has had a long coevolution with ticks. And so the Coxiella genome has been highly reduced over time. It's lacking a lot of genes, but it retains genes encoding for B vitamins and their cofactors, and they provide that to the tick. And so and the tick, of course, provides nutrients for the Coxiella that it can't make on its own. So there's a coevolution. It's a symbiosis, right? Both organisms survive. They don't hurt each other. They take what they need from the other one. It's very common, as we have been learning here. In TWiM, actually, we talk a lot about this. I really like it. And they also say that similar things happen in mice. Modulation of serotonin biosynthesis by the microbiota also happens in mice, although the mechanisms are different. But it's a common point there.
And I'm sure it's probably happening in the human race as well.
I'm sure.
We just haven't figured it out.
And then finally, they say maybe if we manipulate the symbiont metabolism, we can control ticks and other vectors in a similar way, which just goes to show, you get curious about what's happening, and then you end up with some solutions that you weren't looking for. So I just thought this was a lovely, lovely piece of work and a wonderful subject. I love these intricate interactions between, here we have a tick, we have microbes, and of course the mammalian host is playing a role in this as well.
And this paper was from a department that's actually the Human Phenome Institute.
Wow.
I mean, I had to look up what phenome was. But
gone are the days when you have a bacteriology department.
Yes, indeed.
It's too simple a name.
Yes.
My department at Columbia, microbiology, used to be bacteriology.
Yeah, that's a long time ago. That dates you.
Yeah, it dates me. Then it became microbiology. Harry Ginsberg made a microbiology department out of it. And now it's microbiology, and immunology.
So which one of you is having the fire drill?
I have a fire thing going, so I'm going to shut up and let Michael talk for the rest of the time.
All right. So we open the new year with a paper that I believe is posing a Talmudic question, which is, why do antibiotics exist? And this was the title of the paper that appeared in MBio in its most recent issue by Fabrizio Spagnolo and Monaco Trujillo and John Dennehy. And they are respectively from the Queensborough Community College, the Queens College, and the Graduate Center, all of the City University of New York. And I understand from Vincent that they have been previously on TWiM. And so this is the first appearance of them on TWiM. Recall that rabbinical scholars have been debating the finer points of the Talmud for millennia. And I suspect that this was the intent when the concept of these challenging questions for microbiology were raised by our colleague here, Elio Schechter, on his blog, Small Things Considered. For I believe that we'll be discussing the finer points of today's paper for quite some time.
Actually, many of you, quite some time is right, because the paper is long.
It is long. You are correct.
And to be honest about it, I found it verbose.
It's a bit tedious, and many of you are likely aware that the human race is losing its competition in combating antibiotic resistance. Some scholars and those in the know medically have postulated that there is now a serious threat to medicine to move the treatment of infectious diseases back 100 years, if you will, into a or should I say post-antibiotic world. The scary prediction is based on a good deal of evidence that the authors go into, that there now exists high levels of resistance in terms of both frequency and strength that have emerged or have selected for against all clinically approved antibiotics worldwide. And in fact, the infectious disease community has been wrestling with the cold truth that the clinical viability of a newly approved antibiotic, often a billion-dollar investment, is generally less than a decade before its resistance penetrance warrant its rationing, or as the authors offer, guarded usage. So that's the corner that we find the human race that has painted itself into. But we know that microbes have been producing antibiotics for millennia without resistance becoming an existential issue for the microbial world. Hence the question or adventure that the paper is going to address. So namely, why do antibiotics exist? But more importantly, from the microbiologist's perspective, how do we design experiments to validate the arguments in our own answers and our discipline as we begin to contemplate why antibiotics exist? So let's start simple. Let's consider, why do antibiotics exist in nature? First, they've been around for approximately millions of years, and that's not hyperbole. However, in the paper's first figure, the authors offer that from the time that they were discovered by humans and put into clinical use, we have consistently seen resistance evolve to high frequencies in very short periods of time, with the average typically coming out at about a decade or so. From the first antibiotic salvarsan, or arsaphine, an arsenic derivative, one of the microbes, that antibiotic actually targets one of the microbes of the desiratory tract, to glycocycline, which some of you may know as tigracycline, that was developed by Francis Talley, one of the pioneers of antibiotic development at Literally Labs in Pearl River. And I'll actually drop a remembrance that Steve Prozan put into the literature about Talley's contributions to antibiotic development. It's a little bit more fun and much shorter to read. And the bottom line is, antibiotics, they only hang around for, their usefulness only hangs around for about 10 years. But there are some others like the antibiotic present, one of the antibiotics present in the triple antibiotic ointment that many mothers applied to skin knees, polymyxin. It's produced by a bacterium, Pennybacillus polymixa, and it's still a great antibiotic, but it's principally been reserved because it's extremely toxic in people. And if you recall on TWiM, we covered polymyxin resistant or colistin resistance, I think a couple of years ago from the paper about urinary tract infections that was published by Schechter out of Walter Reed's group at Bethesda. So the authors posit 3 questions in helping us wrestle with answering the question, why do antibiotics exist? The first addresses the issue, if resistance is inevitable, and I was so glad they didn't say resistance is futile from the board, why do resistance to all naturally occurring antibiotics not evolve and eliminate the possibility of miracle drugs long before Fleming discovered them almost a century ago? And that's something that, you can see getting dropped into a qualifying exam for a graduate student. Second, and this is really the crux of these authors' arguments, do microbes just manage antibiotics better? Or is there something intrinsic about the microbial use of these wonder drugs that sustains their applicability? And the third question, finally, is why has resistance to even the oldest forms of naturally occurring antibiotics not reached fixation? So the last 2 questions really give you a hint as to what the answer is. The microbes are using antibiotics not as weapons of mass microbial destruction like the human race is using, but rather they're using them for their own purposes, ultimately. And I'll just give you the punchline. As Elio said, the paper is long. The punchline is because the antibiotics are used in a community setting to effectively set the community's composition. And those are the experiments that the authors are encouraging us to develop. And I actually anticipate that the NIH is going to be writing an RFA to actually discuss or to try to figure out if these things are right. So in the remainder of our time, I'm going to actually walk us through how they develop arguments or answers to the 3 questions and helping us decipher the riddle. And I'll throw in some other quips that I think will help us think about this provocative subject area. The authors, as Elio said, provide us with a very nice review of the early days of antibiotics that I'll let the listeners look into on their own, since this is an open access paper. But one of the points I want to address was how they introduced their thesis of their argument that microbes are simply better at managing this precious resource than the human race is. They sort of drop a hint by sneaking in the concept of antibiotic stewardship by the microbes, by offering that in reaction to the antibiotic resistance problem with the treatment options dwindling, public health officials attempted to control antibiotic usage as a means of controlling usage. The hypothesis being the rates of resistance would diminish as antibiotic usage dropped. However, resistance has persisted. And we see every 10 years, once you introduce a drug, it's effectively shot. So resistance has persisted, and the timeline of 10 and done seems to be still holding true, even in the face of such aggressive drug management practices on both small and large scales. The failed approach they offer us to consider is the evolution of resistance by limiting usage is an ecological fallacy. And this is an ecology concept that some of you may be familiar. But for those of you not been acquainted with this term, the term ecological fallacy requires for our discussion to consider that evolution is principally driven or selected for as a consequence of fitness. And fitness is something that we're witnessing firsthand with the SARS-CoV-2 virus as it goes through adapting its variants. The virus is just becoming more fit as the human race responds to this virus. So fitness gains, or simply put, the ability to get your genome transmitted vertically, due, in this particular case of this paper, due to antibiotic resistance, are often offset by fitness costs to the host bearing the resistance phenotypes. And we've talked about fitness costs on TWiM a number of times in the past, but fitness costs to the host bearing resistance phenotypes due to reduced performance in other aspects of their biology. So it's a trade-off. So to put this in perspective, any molecular biologist who has ever tried to maintain their multi-copy high-expression plasmids absent demanding the presence of the selectable marker knows well that if you forget the selectable marker's addition, namely the antibiotic that is required for the resistant trait to be maintained, which then your gene of interest is sort of tucked downstream or upstream, so the gene is maintained and the plasmid is maintained, will be lost. But if you think about it, the flaw in this reasoning has to do with the assumption that the absence of an antibiotic from the environment has the same level of impact, but opposite effect of its presence. In other words, sometimes even if you forget to put the antibiotic in your plasmid containing strain, the requiring you to demand the presence of the marker to maintain the genes, the resistance profile still persists. And many molecular biologists who has forgot to add the tetracycline or kanamycin to their overnight culture can attest that the plasmid can hang around for a few different generations. So, but, bringing this back home, what workers in the field have learned that reversion to sensitivity, going from resistance back to being a sensitive microbe, is neither an immediate nor a necessary outcome of selection simply because a resistant pathogen is no longer in an antibiotic-laden environment. In other words, if you forget, the plasmid may still hang around. This is where their paper gets heavy and makes the designing or thinking about designing experiments challenging. Their explanations for this are subtle and are likely related to the lasting impact that the antibiotic treatment has on the microbe and the community it finds itself in, as well as the challenges and changes at the genetic level that can reconfigure the fitness landscape for resistance populations and make reversion less likely. Bottom lining this, the presence of the clinical concentrations of antibiotics more strongly selects for resistance than the absence of antibiotics selects for reversion to antibiotic sensitivity. But then why does this not help us posit a solution as to why antibiotics exist? However, as they're building their answer to the question of why or how antibiotics have persisted in the microbial world for millennia, they introduce us again, albeit indirectly, to the doctrine of microbial infallibility that we talked about on the last TWiM, namely by introducing us to the concept of the environmental resistome, that a population will maintain some of these genetic traits, which have been demonstrated through the emerging field of in silico microbial genomics, that there exists overwhelming evidence that bacterial genomes have genes that allow them to become resistant to one or more antibiotics through antibiotic resistant genes, and that their precursors, that are found in both pathogenic and non-pathogenic bacteria. Further, this genetic potential that's maintained in all ecological niches where microbial communities are found. And this is where we begin to get the sense that it's all about community. We have to divorce our centuries-old thinking of pure culture microbiology, and we need to go back to what Hungate and others taught us, that it's really all about the microbial community. If you will, the genetic potential serves as a built-in check or police force watching out for looters and opportunists in the microbial community who might use antibiotics to advance their standing, their number, their variety in the community. Now, this is not wild supposition on the author's part, in that they offer that the metagenomic analysis of ancient DNA has observed antibiotic resistance, and that this genetic information predates the use of these wonder molecules before their widespread clinical introduction. So where are the microbes or the microbial communities that support their notion? Well, you can guess where they look. They looked in the dirt. And the soil has been shown to play both a role in antibiotic production, and it also can serve to provide us with deep insight into how microbes have the inherent capacity to inactivate the drugs produced by their communities. So this then raises the next area that may offer us insight into their meta question. And it fundamentally asks, well, what do we know? Well, what do we know about antibiotics in nature is they're not produced at high concentrations or for prolonged periods of times, or in the mega quantities that we find the human race using them, say, for example, in agriculture or even in clinical medicine. Clinically, antibiotics are used to kill or arrest the growth of all susceptible bacteria. If you will, I've already introduced this abbreviation, a weapon of mass microbial destruction, or a WMMD, and any outside consequence, if you will, the meme, too bad, so sad, for the collateral damage inflicted by the consumption of antibiotics is never been really considered when you are trying to treat a life-threatening infection. But the too bad, so sad, we're now appreciating can really have a substantial disturbance to the host microbiome that can last. We've again talked about papers on TWiM that can last for up to 2 years. But what the authors offer is that we should be rethinking the use of antibiotics in the clinic. And this leads me to one of the other things I'm going to drop into the show notes. Did you ever wonder how we came up with the dosing regimen for antibiotics? If you have some time, I would commend that you read an editorial by Dr. Brad Spellberg, who is the chief medical officer at the at the Los Angeles County and the University of Southern California, the LAC and USC Medical Center. And he is also the author of Broken, Bankrupt, and Dying, an interesting read on how we might fix healthcare. And he has been advocating for shorter courses of antibiotic therapy as an approach to help us prolong the utility of this global resource. And the title of his editorial is The Maturing Antibiotic Mantra: Shorter is Still Better. To put his arguments into perspective, I'll simply quote the opening paragraph. The proper duration of antibiotic therapy for various infections is a matter of long-standing consternation. For decades, the standard antibiotic course for most acute bacterial infections has been 7 to 14 days, based largely on the fact that the week has 7 days in it. The reason the week has 7 days in it dates back to an edict issued by Constantine the Great in 321 AD. And so I'm still quoting, to underscore the absurdity of basing 21st century antibiotic course durations on an ancient Roman emperor's decree, he refers to these durations as Constantine units. So one constant unit is a 7-day course of antibiotics, and 2 Constantine units is a 14-day course of antibiotics. However, his cute little argument about Constantine units are indeed backed up by a systematic review from Royer that I'll also drop into the show notes. Shorter versus longer courses of antibiotics for infection in hospitalized patients in a systematic and meta-analysis review where they concluded that shorter courses of antibiotics can be safely utilized in hospitalized patients with common infections, and it's the big ones: pneumonia, urinary tract infections, intra-abdominal infections, and they are able to still achieve clinical and microbiological resolution using shorter courses of antibiotics without adverse effects on mortality, the big one, or reoccurrence of the infection. But let's think about this beyond the notion of 1 or 2 Constantine units. The clinical use of antibiotics is centered upon the concentrated delivery of the compound over time. Now, you have to think about what microbes didn't employ when they were evolving antibiotics. They didn't employ a clinical pharmacologist that was effectively doing the pharmacokinetics and the pharmacodynamics. And the authors give us a great little outset on PK, PD basics, and it's effectively an exercise in clinical pharmacology. I encourage you to read it if you're interested. But at the end of the day, the bacteria and fungi in nature aren't doing this magical PK/PD determination. They're effectively doing something else. So dosing of an antibiotic to cure an infectious disease is indeed dependent upon the PK/PD curve that are generated by the pharmacodynamicists. But the microbe is actually using the antibiotic not as a weapon of mass microbial destruction, but rather to effectively use it to modulate the community. Recall that soil and water don't have either a dedicated liver or a kidney that effectively is the 2 principal organs in the human body that are responsible for getting rid of the antibiotic that's delivered by the medical team. Now, experimental data support this conclusion, showing that antibiotics do indeed persist in soil, water, and even plants over long periods of time before they are degraded by biotic and abiotic forces. However, the ability of microbes to degrade environmental antibiotics suggests that this process is heavily dependent upon, again, the type and size of the microbial communities present, as well as abiotic conditions such as temperature, moisture, and let's not forget pH. pH is very important. Given the reliance on such a diverse and largely uncontrolled variables in the environment, antibiotic degradation is decidedly slow and it's mixed. The most likely result is a longer antibiotic residence time in the environment than we actually see in the clinic. So what else can nature offer us over the clinic in trying to understand how to get at an answer? Why do antibiotics exist? And they again tumble to the fact that antibiotics are often signaling molecules that effectively affect transcription of unique operons in the microbes. And the consequence is that it effectively is serving to change the community. So if concentration, duration, and scale of antibiotic use in nature are so vastly different from what we're using in the clinic, it shouldn't be any surprise to us that we only get 10 years and then we're done. The question, what we need is, how can we, the human race that desperately needs antibiotics to treat life-threatening infections, how can we learn from the microbes to effectively use the antibiotics better? And they actually introduce a neat concept referring to how antibiotics are used in nature as a Rosetta Stone, effectively illustrating how we can think about antibiotic use in nature to give us an inkling of what's actually going on. In the end, they offer us that humans broke the microbial social contract by co-opting antibiotics under the presumption that they were an underutilized tool, transforming them into a biological weapon of mass destruction. It was our own hubris that effectively has really cratered us. And so in doing so, the human race created a potent selection for higher and higher resistance levels, losing the ability to control or manipulate. So it was inevitable, if not foreseen. So in answering the question, why do antibiotics exist, they end by challenging us to demonstrate through experiments that antibiotic resistance is not an evolutionary reaction to antibiotic warfare, but rather a component of the preferred assembly mechanism for microbial communities. Now, the reason I'm starting off the new year like this is to really think about using antibiotics to decipher the interactions going on with the microbiome and to understand how antibiotic perturbations can really alter that community. And we've seen many microbiome studies that read like reading the New York telephone directory, where we see all of these species and then we see fancy statistics, and we really struggle trying to figure out what's going on. What we need are thorough and thoughtful experiments using antibiotics as tools to understand how to assemble the ideal microbial community to help us that will actually have a positive impact on human health that will then impact human health. And as the authors end their story, impact human health for lifetimes to come. So this is a bit provocative. It's a bit different than our normal fare, but I thought it was a good way to start the year, forcing us all to think a little bit. And as Elio said, it's an interesting place. It's a little bit long-winded, but it was a fascinating read. And I read it on a plane, so I was a captive audience.
I like this idea of hubris. I think for all infectious diseases, they're a result of our hubris.
Yes.
Right? We invade ecological areas where other animals exist. We get their viruses. We start to do
habitat destruction,
growing animals for food. We get their viruses, and we thought that resistance. I think there's a good point for students in particular. Resistance is not an evolutionary thing. It's always been there.
Yeah. Well, one of the things I didn't go into is they have a bit of a discussion about using antibiotics to decrease the farm to table time. Yeah. They figured out that very early on by just putting a little bit of antibiotic into the feed.
Right.
You put on more weight per unit time than not. And, that antibiotic in animal production is really one of the issues that we have with dumping lots of antibiotics into the environment. And, it's really, it comes down to hubris. We don't understand the tool before we begin to use it. I think my father said, if everything, what was it? It's something about a hammer and a nail. If everything looks like a nail, you use a hammer.
That's right.
And
that's right.
it's our own hubris that I think has gotten us into this pickle that
Indeed.
we're in.
Yeah. We think we can take over everything.
Yeah.
But isn't it going to be very difficult to introduce ecological and evolutionary thinking into the practice of medicine? Isn't it going to be tough?
It's going to be tough. And
I agree.
And that's why I think we need to consider using organoids in which we, tinker with the microbial community that's established in the organoid, add a clinical course of antibiotics, like Vincent showed with the paper on tryptophan and the Coxsackie symbiote. We are developing sufficient expertise in deciphering these complex situations. And, everybody is on the bandwagon of the microbiome. And what many people are doing is just collecting the New York telephone directory.
That's right.
They're not really trying to develop the mechanism of understanding what's going on. And at the end of the day, you may have a great probiotic, but the minute I take an antibiotic, all bets are off.
Right.
So, I think you were the one, Elio, who said, okay, you've given me the New York telephone directory. What do I do with it?
That's right.
Who am I going to call?
That's right. That's a great point. I like this. I think it was a very nice way to start the year. So thank you, Michael.
Well, thank you, everyone.
And that'll do it for TWiM number 258. You can find the show notes at microbe.tv/TWiM. If you like what we do, consider supporting us financially. We'd love your support. You can go to microbe.tv/contribute. There's several ways you can help out. And we are a 501c3 corporation, so your contributions are federal tax deductible. And, of course, if you have questions or comments, send them to twim@microbe.tv. Elio Schechter is at Small Things Considered, a lovely blog that you should go check out. Thank you, Elio.
My pleasure. Thank you.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone. Happy New Year again.
And I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Microbiology for their support of TWiM and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.